Este encendería. Ah. No, es que estaba apagado. Empiezo desde el principio. Vale, perfecto. Bueno, buenas tardes a todos los que estáis al otro lado de la pantalla en este viernes previo a las vacaciones de Semana Santa. Y tenemos hoy la suerte de contar con una persona que es Jimena Baleriola Gómez de Pablos. Ella se formó en bioquímica en la Universidad Complutense de Madrid. Realizó su tesis doctoral sobre la integridad de ADN y cómo se puede afectar por diferentes alteraciones moleculares y el impacto que puede tener esto sobre la célula y la muerte celular programada y en relación con el desarrollo del sistema nervioso central. Un tema que a mí personalmente me parece complejo, pero creo que ella nos lo va a facilitar. Posteriormente se trasladó a la Universidad de Columbia en Nueva York y allí trabajó sobre la enfermedad de Alzheimer, también sobre mecanismos celulares que en el fondo lo que hacen es intentar comunicar un tipo de células del sistema nervioso que son las que le dan soporte con las células propiamente neuronales. Hay un sistema de comunicación que nos explicará ella con más detalle y creo que a mí no me corresponde. Estando allí fue candidata, solamente hay 12 candidatos, ella fue uno de ellos al premio Blackney y posteriormente, que es un premio que se da a jóvenes científicos, evidentemente que sobresalen sobre el resto. Y posteriormente ha trabajado en neurodegeneración nerviosa y ha estado con becas de Ramón y Cajal, que son becas muy competitivas. Posteriormente estuvo como Basque Fellow aquí y ya desde este año está como Basque, digamos, como investigador ya consolidado en el Centro Achucarro de Neurociencias. Ella trabaja en neurodegeneración que es una enfermedad que todos sabéis que está cada vez más en boga por su frecuencia y porque se está trabajando mucho en frenar la evolución de la enfermedad de Alzheimer o de otras enfermedades neurodegenerativas, y trabaja sobre todo en esta conexión que hay entre las células gliales de soporte de las células más nobles cerebrales nerviosas en un sistema de comunicación que hace que se produzcan más o menos proteínas. Y sin más, os voy a dejar con Jimena. El título de su charla, esto sí que lo voy a leer en inglés, que es Remote Control of Gene Expression in Neurons. Cuando quieras, Jimena.
Thank you very much to all of you. So this talk is going to be held in English. And thank you for the kind introduction, and thank you to all of you that are on the other side of the screen. I'm going to simplify my talk a little bit today because we work in a very complex topic, and I want you to really understand what we do in the lab, why we do what we do in the lab, and why that could impact neurodegenerative diseases. So essentially, first I'm going to introduce myself, and I'm going to introduce my group. And then I'm going to talk to you about basic knowledge of gene expression regulation. And then I'm going to talk to you about what part of gene expression regulation is the one that we studied, and how can glial cells can influence the neurons in this kind of processes. So, as I already told you, we work in the Achucarro Basque Center for Neuroscience. This is our building, which is located we are located in the 3rd floor of this building. This building is in the Laude Science Campus of the University of the Basque Country. This is part of the Bizkaia Science and Technology Park. And we are a group of all women. So for some reason, our lab is an all-women lab. Not that I looked for it, it's just the way it worked. So essentially, my lab is composed by 4 very talented young pre-doctoral researchers: Maite, Maria, Aida, Yosune, and that's me, okay? And then also I want to introduce Adara, who's another pre-doctoral researcher that, although she's not located in my lab, she's in this other lab, also at Achucarro, the neurobiology lab. I co-mentor her PhD thesis. So she's also interested in part of what we study in the lab, essentially. So that's why it's worth mentioning that Adara also is interested in what we do. And she sometimes comes to do the experiments to our lab over here. So that's important. So essentially, we're interested in the regulation of gene expression, but it's from a very special point of view. But first of all, I'm going to explain to you what gene expression is. Essentially, gene expression is the conversion of your genes to a protein, because at the end, the proteins are the macromolecules that elicit the function of the cell. So proteins are the building blocks of our neurons structurally. So the membranes are made out of proteins. The skeleton of the cell is made out of proteins. Also the enzymes that are involved in our metabolism are essentially proteins. Hormones that communicate one cell with the other are also proteins or peptides. So at the end of the day, the protein is the functional manifestation of our genome, okay? So in order for our genome or our genes to become a protein, the DNA has to go into different steps. So first, there's the conversion of the DNA to RNA within the nucleus. That is through a step called transcription. So first, the RNA that is going to be transcribed is going to be immature. And because of that, it needs to be processed to make a mature RNA. So in the cells, we have a lot of different kinds of RNAs. But today, I'm only going to talk about the mRNAs, because these mRNAs or messenger RNAs are the ones that encode the proteins. So only these ones are going to be converted into protein. So after the mRNA is made, this one exits the nucleus and undergoes what is known as translation. Essentially, translation would be the conversion of the RNA to the protein. And similar to what happens with the RNA, proteins in the first steps of synthesis, they're going to be immature. And so they have to undergo processing in order to get a mature and active protein. So essentially, my lab is interested in the step of conversion of mRNA to protein. But before I go into this details of this translation step, which is the one that we study, I also want you to take into account that these different steps have to be tightly regulated in eukaryotic cells. Well, eukaryotic cells or any other cell to that matter, because no two different kinds of cells, like for instance, liver cells or brain cells, they do not necessarily have the same repertoire of proteins. Actually, what makes a liver cell is the fact that it expresses proteins that are able to clean from toxins your body. Whereas neurons, for instance, it's the role of neurons, it's to elicit neurotransmission. So essentially transmit information within brain cells and from the brain to other regions of the body. So in order for brain cells and for liver cells to be different, these cells have to regulate their gene expression tightly, turning off and turning off on different genes, okay? So this would be an example. If you go to a bar and you've drunk a lot of calimocho one night, your liver cells have to express a lot of alcohol dehydrogenase because you need to clean away the alcohol that you've drunk the night before. So this gene is going to be turned on. Whereas, for instance, liver cells don't need neurotransmitters. So in this particular case, these genes are going to be turned off. Essentially, that means that this protein is not going to be expressed. Whereas in the case of the neurons, you're going to have neurotransmitters, but you're not going to express the proteins that are involved in the clearance of the toxins from your body. This is something that it's studied by other people, but as I told you before, our lab is more interested in this step over here. So I'm not going to go into the details of the mechanisms that regulate this step of transforming the DNA to a particular RNA that would eventually be transformed into a protein, but rather the step of converting the mRNA to protein. So during the talk, I'm going to talk indistinctively about translation or protein synthesis. They're different steps of the same transformation step from RNA to protein. Okay, they're different, but in order to simplify, I will talk indistinctly about translation or protein synthesis. Now, when you Google a protein synthesis in eukaryotes, what you find normally is cartoons that are similar to this one, right? In which eukaryotic cells are essentially depicted like round structures, okay, with no distinctive, structural complexity of any kind. And again, they're depicted with a nucleus that has the genes, and then genes are transcribed into mRNA, and then the mRNAs exit the nucleus, and they're processed and transformed into protein. However, the reality tells us that proteins are not as simple as this, but they're rather much more complex. This is actually one image that we took under our microscope from an oligodendrocyte. This is an astrocyte. This is a brain cell. And you can imagine that if cells are this complex, most likely these processes are going to be more complex than what they're normally depicted when you Google them. So as I told you before, proteins are the functional, let's say, macromolecules or the functional manifestation of our genes. And as I told you before, they play different roles, including structural roles. So this is the structure of an astrocyte, okay? And you can see how it has a soma that actually, it's very similar to all this thing. And then it has all these processes that we say, these processes. Obviously, the entire process needs to have proteins, and proteins need to be transported somehow over here. And the way normally you see the cells that are drawn in textbooks or when you Google them, it seems like all the machinery that implies transcription and also translation are sitting like in the center of the cell, right? So essentially and that's what the textbooks tell us, and I'll now show you. So essentially, from this complexity, we might think that the proteins that are generated here somehow have to travel to the periphery of the cell. Again, if you now Google protein transport in eukaryotes, things look sort of like this. Okay, I'm happier with this because the cell is more complex. It's not like a round shape entity, which actually almost does not exist in our body. So, but something more like a skin cell or an epithelial cell or something like that, that has certain polarity, certain asymmetry. However, in this kind of cartoons, you can also see how normally they depict protein synthesis to be a process that happens close to the nucleus, right? Here we've got new protein, it's close to the nucleus. Also, this protein is close to the nucleus. And then those proteins need to get to different target compartments. The target compartments is where they normally elicit their function. So a mitochondrial protein will be transported to the mitochondria. A nuclear protein will be transported to the nucleus. And then you've got some membrane proteins that would go to the membrane, hormones that would be secreted to the outer space and then be transported to other cells, etc. But this is also not reality. I mean, it is reality, but there's an alternative reality as well and complementary. And that's actually what we study. So again, neurons are more complex than what you see. So this is actually a neuron. In the neuron, you would find a soma, which is similar to this structure over here, but you've got these branches that come out from the neurons. These are called the dendrites, and this is the huge axon. The difference between the dendrites and the axons is that the dendrites are normally the receptors of the neurotransmitters, whereas the axon is the road through which the neurotransmitters travel, and they release the information from one cell to the other. So essentially, axon is going to release the information, the dendrite is going to receive the information, and that's how you get communication between the neurons in your brain. So you see this huge complexity that you have within a neuron. And actually the fact that you've got dendrites that play one particular role and axons that play another role already tell us that the proteins that have to be distributed in the dendrites are most likely, in a lot of cases, different to the ones that are sorted within the axon. And the way this cartoon looks or tells you that protein synthesis of axonal proteins and neurotransmitters happens is, again, in this domain over here, close to the nucleus, you've got protein synthesis. Those proteins are going to mature through this Golgi complex, okay? And they're going to travel all the way through the axon until they get to the axon tip. That's fine, that happens, okay? But it's not all that happens. So that's what the dogma says, right? And for a lot of years, the scientists, cell biologists in particular, thought that maybe this was a little bit odd to be the only mechanism for distributing proteins within a neuron. And the reason is the distance that you can find between the cell body and the tip of the axon. So if you look at a motor neuron, for instance, in which you've got here the cell body in the nucleus, so theoretically you would have here the genes, and then the genes will be translated into RNA, converted into RNA, the RNA would exit the nucleus, and then proteins would be synthesized over here and then transported there, okay? If you think about an axon of a motor neuron, this axon can actually measure, depending on how high you are, around 1 meter or more, okay? If you put that into microscopic distances, imagine that this baseball would be the soma, then the axon would be more than 1 kilometer. The tip of the axon would be more than 1 kilometer away from the soma. So this was actually very puzzling for a lot of neuroscientists. And they measured what was actually the pace or the rate of transport of different kinds of proteins from the soma to the axon to see if actually protein transport would be the only mechanism to make a protein to reach the cell body, from the cell body to reach the axon from the cell body, essentially. And this is an actual experiment in which the scientists, what they did is to feed radioactive amino acids. So essentially, they would be able to measure new synthesis of proteins by measuring radioactivity. And they measure the time that it took for these proteins to reach the tip of the axon. And if you think about a 30 millimeter axon, it would take 16 days for this particular proteins to reach the tip of the axon. That's a very slow pace. If you think about a 1 meter axon, like in the case of the most extreme motor neurons, if you think about fast transport, okay, it would take maybe 10 days or whatever. But if you think about slow transport, in some cases, some proteins like neurofilaments and microtubules could take 1,000 days to reach the tip of the axon. So that made scientists think that maybe axons, and we know that happens not only in the case of axons, but also in the case of dendrites, maybe we're able to autonomously regulate their protein production so that they would have an autonomous or semi-autonomous metabolism, and they would be able to synthesize proteins without fully relying on the cell body. Obviously, they have to rely on the genes, right? But without fully relying on the cell body, and that I explained. And actually, that is true. And I'll show you some experiments showing that is true. But essentially what those people hypothesized so many years ago, because this is a hypothesis from the 19th century, means what it tells us is that there's probably two ways of transport of proteins within our cells. So it is true that actually proteins can be synthesized in the soma, close to the nucleus, and then be transported to the periphery of the cells. But it is also true, and has been seen in many different cell types, that there's also another mechanism in which you do not transport the protein, you transport the RNA. And once the protein of that RNA is needed, then the protein is locally produced at the periphery. Okay, so it's not transport of protein, it's transport of RNA. This is called local protein synthesis, and it's this what we study. So local protein synthesis is not something fancy. So in neurons, you can think about the distance problem, and you can think, okay, it makes sense. But actually, RNA localization and local protein synthesis has been seen in every polarized cell. But I told you, remember, cells are normally not spherical, they're not rounded, they're normally very asymmetrical. So because they're asymmetrical, their proteins have to be asymmetrically distributed. And for asymmetric distribution of proteins, actually a lot of cell types, including yeast and including bacteria, they do protein synthesis locally. They're able to synthesize proteins locally. So they would transport the RNA and not the protein, and then produce the protein at target sites. So this is experiments from our own laboratory. I'm not going to go into technical details, just to show you the autonomous ability of axons to make their own proteins. These axons have been, so this is a way that we have to culture the neurons, okay? And here we see the neurons in our culture plate, and then you've got the axons growing through a membrane that is semi-permeable. And then you've got the axons, they're axons and dendrites, but mainly axons, on the other side of this membrane, right? Forget about the glia for the moment. I'll talk about the glia later. So what we can do in this kind of culture is essentially to cut the soma, take away the soma, and keep only the neurites, okay? And then after a while, we're able to measure protein synthesis in the axons without the cell body being there. So if you're able to measure proteins in the axon without the soma being there, that means that that protein synthesis is actually local, because you don't need the component of the soma. And this is actually what we see in this particular case, okay? So this stain here that is a black schmutz or whatever it is, this is actually new proteins that are being synthesized during 30 minutes that we measured protein synthesis. And you can see how there's a huge increase in this particular case, okay? So in this particular condition, we can tell that there's protein synthesis going on in the axon that is independent of the soma. So essentially, what that tells us is that this dogma that proteins are synthesized in the soma and then are transported to the axon is not always true, because when you cut the axon away, you are still able to measure proteins here, newly synthesized proteins. So originally, protein synthesis, local protein synthesis in neurons was first described in dendrites, because apparently it was easier to study it in dendrites. I'm not going to go into the technicalities of why it's easier to measure them in dendrites, but that's the case. And intra-axonal protein synthesis, which is the one I'm more interested in, was demonstrated many years later. I'm going to give you a brief story of how the field has evolved over the years. You don't need to understand the titles and whatnot of the papers. It's just so I can show you an evolution of the field, because this is a field that not a lot of people study, which is actually good, but it's also bad. It's good because you don't have a lot of competition. It's bad because sometimes they don't believe you, whatever. So as I told you before, in the 19th century, it was also already suggested that axons were able to produce proteins locally. But in the case of vertebrates, we had to go until 1943 to detect RNAs in axons, in the case of vertebrates, by absorption microscopy. And also we had to wait several years to detect ribonucleoproteins or de novo protein synthesis. And in the meantime, other experiments had failed. Actually, we've had up and downs in the field. Sometimes there was some evidence that said, yes, axons can synthesize proteins, and then suddenly you had another paper saying, no, we could not see that, etc., etc. thankfully, techniques have evolved, and now we have better techniques in order to detect this event. So axonal protein synthesis, see, 1960 was the first time it was really measured. And then several years had to pass in order to see polyribosomes, because obviously you need ribosomes in order to synthesize proteins. That's the machinery where proteins are being made. And then the first axonal RNA was identified not very many years ago, which is the one of beta-actin, which is part of the cytoskeleton of the cell. And then from here, the field bloomed a little bit, okay? See? Still, some papers, this is when I started my postdoc and I moved to New York. Some people were saying that this event was actually not true. And this is when I started my postdoc training. So it was a field that actually was blooming at that time, and still not nowadays, not very many people study it. What I did at that time is to study local protein synthesis in axons in the context of neurodegenerative diseases, in particular in the context of Alzheimer's disease. And at that time, we were pioneer in the field, because people studied local protein synthesis in other contexts, for instance, during embryonic development or in injured nerves, but in the case of peripheral nerves, not in the central nervous system. So we were the first to describe how local protein synthesis could play a role in Alzheimer's disease. And that is after that is when I came to the Basque country. Why was for us important to study this event in the context of a neurodegenerative disease? Because in Alzheimer's disease and in other neurodegenerative diseases, projections are affected. So axons and dendrites are the ones that are affected. And in this particular case, for instance, there's the theory of the basal forebrain hypothesis, or of the cholinergic hypothesis in Alzheimer's disease, which is still not super clear, but there's a fact. And the fact is that one of the earliest signs in Alzheimer's disease is degeneration of basal forebrain neurons, whereas in this particular case, there's no amyloid plaques. I'm going to tell you what amyloid plaques are. Amyloid plaques are these things here, which are one of the main triggers and one of the main hallmarks of Alzheimer's disease. Also, maybe not the plaques, but the peptides, soluble oligomers of beta amyloid are supposed to be the triggers of Alzheimer's disease, together with these tangles over here made of tau, okay? So when you diagnose Alzheimer's disease, and until actually now almost you've got a postmortem diagnosis, what you see normally is these plaques and these tangles, right? So one of the things is that when you open up a brain postmortem, you see that this region here of the brain degenerates. There's cells are atrophied, and there's actually cholinergic dysfunction early on in patients. However, you don't find plaques. Plaques are normally essentially found in the cortex, right? But what is the characteristics of these cells here? These cells project to the regions of the brain that have plaques, which means that amyloid is affecting the projections. So what we saw is that actually when you inject amyloid into the hippocampus or into the brain of a mouse, you are able to induce local protein synthesis in axons. And that local protein synthesis is required for the cells to die. So if you block local protein synthesis, you rescue the cells from dying. So that's why, for us, it's important to look at this event. This is one of the experiments that also my students did in the lab to demonstrate that amyloid peptides are able to induce local protein synthesis in axons. Also, I don't want you to understand this is just a pretty picture. Hopefully you find it pretty too. So here what we did is to culture neurons and target these neurons with beta oligomers that are known to induce cell death in the Alzheimer's brain. And here in green, we use a chemical called cytoRNA select that labels the RNA. And you can see this is the axon because we take this particular, we stretch it, and you can see how you've got RNA far away from the cell body. The cell body will be here, and then here you've got the tip of the axon would be over there, sorry. And then you also have newly synthesized proteins that are here measured with this particular chemical, puromycin, that is here we detect in green. So whenever you see colocalization between these two events, which are the yellow events, hopefully you see them better here in cyan, these are actively translating RNAs that are being translated far away from the cell body, so in the axon. You can maybe see them better here, and this would be a control treatment, and this is the amyloid beta treatment. So as you can see, amyloid peptides are able to induce translation within the axon. So this is one of the things that we do. We continue to do in the lab, but we are making things more complicated. They were already complicated, but we like things that are complicated. Why not? So as I already told you, other interest in our lab is to see how glial cells are able to regulate this event. And why is that? Because it's been already shown that glial cells, which are here stained with this, here in this greenish kind of thing, okay? You've got here the axon that is stained with beta-actin, right? These cells, which are a kind of glia called Schwann cells that are the one that myelinate the peripheral nerves, okay, are able to transfer RNA to the axons, in particular to lesioned axons. When you make a lesion to the axon, you transect an axon. These cells over here are able to transfer actively RNA to the axon, which means that maybe there's a regulatory mechanism in which local translation in axons is not fully cell autonomously regulated, but that glial cells might be participating in this event. And we're interested in seeing what is the contribution of these glial cells in the context of Alzheimer's disease. So our hypothesis is that glial cells are able to release one kind of vesicles, which are known as extracellular vesicles, because they're vesicular structures that have packages of information that are secreted to the outside. And those vesicles are now uptaken by the neurons, and those vesicles within the neurons are able to regulate translation. And this is actually what depicts these vesicles, okay? This is also a vesicle, and this is something that has been hypothesized also some years ago, but not a lot of people study. So essentially the answer to our question of whether glial cells are able to control translation in neurons by releasing extracellular vesicles, for us the answer is yes, and I'll show you another experiment that we have in the lab. This is more illustrative, nothing about results. This would be the basal translation that you get in an axon. This would be the cell body, okay? The brighter the color, the more translation that you have, okay? So essentially we are able to co-culture neurons together with astrocytes and see how those affect translation, or isolate the extracellular vesicles that are released by this cell type, put them into neurons, and we see translation going up. So far, we do see that this way of communication regulates translation in neurons, local translation in neurons. So now what we're trying to see, and hopefully other people we would like to know, is what is the consequence of this phenomenon in Alzheimer's disease, either in the progression or in the slowing down Alzheimer's and other neurodegenerative diseases. So I don't want to go into more detail because I could go into more complex things, but it's Friday. So just to conclude, I hope I've convinced you that besides transcription and translation, RNA transport and local translation should also be included in these cartoons that depict gene expression regulation, because we think it's an important event, at least in neurons. And I hope I convinced you. Also, we know that RNA localization and local translation are conserved mechanism in eukaryotes. We still don't know what is the full advantage of having local translation, because I have to tell you that proteins that are locally produced, they're also globally produced in the soma and can be transported. But for some reason, cells rely differently on local versus global production, and we still don't know why. Nobody knows exactly why. And also we know that local translation in neurons, and that's something that more and more people are studying, is involved in neurodegenerative diseases. And I hope I also convince you, although this is work in progress, that's what we do in the lab, that local translation can be remotely controlled by non-neuronal cells by the release of extracellular vesicles. So thank again to the people in my lab, all the PhD students and the funding agencies that pay our salaries, and also funding agencies that fund our research. And that's it.
Sorry. So,
it seems that the triggers are both the aggregates of amyloid, that for some reason amyloid aggregates buildups in the cell. It is thought that the first buildup occurs intraneuronal, although the hallmark postmortem is extraneuronal plaques. That's one of the things, but also tauologists would think that actually the main trigger would be hyperphosphorylation or misfunctioning of tau. Other people think it's both. In my case, I think amyloid goes first. So essentially the buildup of amyloid would start to alter cellular functions in general, and that misfunction actually starts at the level of the synapse. That's why for us it's important local protein synthesis, because at the level of the synapse, where you've got the tip of the axon and the tip of the dendrite, there's local protein synthesis going on there. What is causing that? It is not really known. So people normally study the effects of amyloid on synapse, but I mean, there's a lot of mechanisms that are involved in this dysfunction, but I think nobody really knows.
so we have to wait for
Yeah.
the knowledge of that.
Yes, actually, all the therapies that are based on trying to clear amyloid are not working. Tau therapies are not working. The only therapy, or one of the only therapies that you would have in the market, is a substitution therapy in which you essentially try to put a chat, acetylcholine transferasa, bueno, that one, which is because one of the first hallmarks, it's a cholinergic dysfunction, and that also does not work. So it's not like Parkinson that you put levodopa and then you restitute the levodopa. Here they try to do the same and it doesn't work. So probably there's something that we're missing that is still not known. In my case, I think that we are catching Alzheimer's too late. And whenever the changes are made, it's already too late. So, but we still don't know what comes first.
Yeah, are all the dementia we know now are similar?
No. No, also because different regions of the brain are going to be differently affected. Means there's, I think, for instance, that a lot of neurodegenerative diseases, they share, in some cases, that they're synaptopathies, so that they affect the synapse first, obviously from different kind of neurons. And that's why I want to study from the point of view of local protein synthesis, because my gist is that maybe synaptopathies are accompanied by changes in the local proteome, right? But no, they're not the same. Actually, there's problems even in diagnosing Alzheimer's disease, because sometimes you misinterpret other dementias, think that it's Alzheimer's and it's really not Alzheimer's. So it's a little bit tricky. Neurologists sometimes, at least when I went to some meetings of Alzheimer's and Parkinson's, there was a lot of pathologies that were complaining, because in the sense that, you know, you've got all these therapies trying to figure out how to cure Alzheimer's, and yet the etiology of Alzheimer's is not fully known. So, but that happens with other dementias as well. Problem is now we live much longer. So we mix up things, I think.
I think you have been very clear for all the students we have at the other side of the screen, because there's there are no questions. So maybe it is because it is Friday and they are very tired after 1 week of study.
Sí,
podéis preguntar en
que si
castellano.
no, a lo mejor se cortan por eso también.
Puedo entender que el inglés técnico sea complicado. O sea que si queréis hacer alguna pregunta en castellano.
Sino que me escriban también. Que por eso he puesto el mail.
Sentiros libres de hacerlo. ¿Nada? Ya, ahora hay diferentes estudios por parte de neurología con la enfermedad de Alzheimer, que lo que intentan es, como tú has dicho previamente, retrasar la enfermedad. Pero realmente, en lo que tú sepas, aunque ya sé que trabajas en investigación básica, ¿hay realmente algún tipo de tratamiento que funciona?
No. Por eso, porque no es como el Parkinson.
Y lo que mejor funciona realmente es educación para mantener un poco activa la memoria.
A ver, yo no es que yo no crea en esas cosas, pero tampoco me he leído artículos en ese aspecto, ¿no?
Bueno,
claramente sí que se hace ese tipo de terapia, ¿no?
ajá.
Pero también se ha visto que la actividad neuronal en otros, es que es un poco contradictorio, también se ha visto que la actividad neuronal al final es mala incluso para el Alzheimer. Entonces, claro, ¿qué es lo bueno?
Bueno, aquí tenemos una pregunta.
Muy bien.
Es, what led you to want to study gene expressions?
I've always done sort of cellular molecular biology, and it was not gene expression exactly what interested me. It was actually this particular mechanism that leads to gene expression, because I didn't know it existed. I didn't know axons had RNAs. So the first time I contacted my boss in New York, I saw that he was working on this, and I said, wow, this is very strange. I don't think anybody knows that axons have RNAs, because that goes against the dogma completely. You know, so I was curious about that. Why are RNAs in the axons? What are they doing?
Yeah.
So it's that aspect of gene expression regulation that interested me.
Okay. No more questions by the moment.
It's okay.
You need the microphone, Jacqueline?
Your investigation is
it's basic science, but we try always to translate it to diseases. There's different reasons for that. I was trained in developmental biology, and the way I was trained is that developmental biology, even if it's basic science, it's useful to understand mechanisms of dynamic neuron. Neurons are super dynamic during development. And after a while, especially axons are super dynamic, but after a while, people think that they sort of stabilize and they're not so dynamic anymore. But when you've got diseases, some of the dynamic mechanisms that you have during development, they try to reactivate. Okay, because that's a changing environment. So we thought that maybe trying to understand basic mechanisms during development would help us understand basic mechanisms of disease. And my boss in New York had the same idea. He was also a developmental biologist. And we thought, why not apply the knowledge of development to disease? So it's sort of basic science, but we always try to apply it to diseases. Actually, part of the funding that I have is from the Alzheimer's Association. And they do translational, they do basic science, but also always from the Alzheimer's point of view. So it's in the middle, yes.
Young people, maybe they don't know this difference between basic science and applied science.
Yeah, so for instance, when you study in development, how nerves grow and how the brain is formed, that is, you study basic mechanisms that you need in order to make a brain, in order to make a liver, be a liver, etc. But then if you study it in another context, like for instance, when you put a stimulus like we do, like amyloid is bad for neurons and it's going to be involved in Alzheimer's disease. If you study mechanisms in that context, that's going to be more disease related. Although you most likely not gonna have direct applicability because you're not discovering drugs, but you're discovering pathways that you might apply drugs to. So that is the preclinical part.
So I think we can finish now because there are no more questions.
That's okay.
Thank you.
Thank you. Thank you for inviting me.
Very interesting.
Thank you.